Okay, good morning. So we'll talk now about how to generate the backbone for TVB. So far we heard a lot about theory, what TVB can be used for. You had some experience with the GUI and the Jupyter Notebook, but now we will focus on the whole work that has been done or has to be done before we can actually go to the step where we're simulating with TVB. Yeah? So you were using the default connectome data that was provided with TVB, or we gave you, but now we want to know, okay, what do we have to do to actually get this data before we can run a brain network simulation with the virtual brain. So, does it work? Yes, if I turn it on. So basically the question is how to get from raw imaging data of the brain to such a network model. Yeah? So we might have many different modalities we can scan. We have structural scans here, T1 weighted, T2 weighted, proton density. We have fMRI images measuring the activity of the brain over time. We can measure, we also have field maps, for example, to measure the magnetic distortions in the image, and diffusion-weighted images which measure the diffusion of water molecules inside the brain, which we will use to generate streamlines or fiber tracks inside the brain. We try to approximate this way the connection between different brain areas. And in some cases, we also use PET, positron emission tomography, for example, in Alzheimer's disease, tracing amyloid or tau proteins. So all these different modalities we want to combine into one model. Yeah? And this is me, so what I want to show here that all this data is individual, individually for a subject. So this is all, no, well, that's not. But the rest is taken from my brain. Now, what we want to have is, in the end, a 3D graph of the brain. So we have nodes here, right, and the edges connecting them. And these nodes correspond to different brain regions, right? So you have to use some kind of division or some kind of brain atlas to divide the cortical surface and also subcortical regions into functionally different brain areas. So this is one aspect, the brain atlas and the parcellation, and the second one is the connectivity strengths. Yeah, how do we estimate this? And then there's a little bit more if you want to go further, or if you want to run more advanced simulations with TVB. But this is the main part, right? So very briefly. If we take my structural scan here of the brain, what do these preprocessing algorithms and pipelines really do is, well, they strip away everything that is, so they do artifact correction, that is a very important step we'll cover. Then they strip away everything that is not the brain, because this is our main focus. Then we do tissue segmentation. We have algorithms which can tell us, okay, where is white matter, where is gray matter. This is specifically important for constructing the connectome, for constructing the brain atlas as well on the cortical surface, subcortically, and also for streamlined tractography later on. We add subcortical regions. And we have here cerebellum, thalamus, putamen, amygdala, hippocampus, and so on and so forth. So here we are using basically 19 different subcortical regions. I guess this is for, I'm looking at the DBS guys, this is probably not sufficient for your purposes. When you are considering DBS targets, you want probably have a much finer grained division of the subcortical areas. For TVB, this shouldn't be a problem as long as you can manage it technically to include such a segmentation into the subcortical area and track the streamlines still between them, or come up with some kind of measure, some kind of way to determine the connection strengths between these areas. So the basic principle that still applies. You just would then use another atlas or another type of subcortical segmentation. For this is what we mainly use here, this more coarser-grained subcortical segmentation, but we then have also a parcellation on the cortical surface. So this is what you see here, right? The gray matter is now divided, depending on the color, into different brain areas, and right, these give the they are the initial start for generating the connectome. If we then also track the fibers between them, what we do is we group fibers together, which go into one area and come out of it to create a 3D network. So as a general overview, here I'm taking a screenshot or I'm taking an image from a pipeline constructed here by Michael Schöner. You already saw him on Monday, and he'll talk today later on as well. And I just want to say that there are many different pipelines out there with for many different purposes designed. What TVB really cares about in the end is this, right? The structural connectivity, a region mapping for the cortical surface, like this, yeah, a subdivision of the vertices on the surface into different areas. Maybe even a functional connectivity, because many people use then functional data to fit the simulations too, to fit simulated and empirical data, to infer some kind of, to make inference on some parameters, and EEG as well as an option. So in this pipeline, EEG was also used. We have empirical recorded EEG, and we can also use our structural data. So we know the cortical surface, which we use as the position of the dipoles. So we consider dipoles on the cortical surface, which then lead to an electrical field that we can actually measure with EEG sensors on the cortical surface. So there's we use forward modeling. There's different methods for this. I'll briefly discuss this in the end of the talk as well. But what I want to say is this is a general overview. We have diffusion MRI data where we estimate these glyphs in each voxel of the brain. These glyphs basically represent the probability of fibers in each voxel. Yeah? So these big lobes indicate, okay, in this direction there is a huge probability that there's a fiber transversing this way. Small lobes are a small probability. And this information can then be used to, in an iterative process, to walk through the brain and approximate fiber tracks, as you can see here. And together with the structural image, with the tissue segmentation and cortical parcellation, we generate the structural connectome. Bold data, similar. We map the bold data to the regions identified in the structural image. There's aligning. There's one big step is aligning the different modalities to each other. So we can extract region-wise time series and well then can do whatever we want with this. But what we usually do is we do these pairwise correlation coefficients to construct a functional connectome. And yeah, EEG, leaf field matrix. Many different pipelines out there. This is just one example. I'll cover other aspects later on. So just the outlook for this talk. We had the pipeline overview already. So this was the beginning. Just to give you an idea of what we want to do. We will briefly cover then structural preprocessing. See what kind of artifacts are there. What are the main steps in structural preprocessing? What information do we want to take out of there? Diffusion preprocessing for this streamlined tractography, functional preprocessing. We will also talk about the cortical parcellation. How do we define? How do we identify functional different units on the cortical surface? There are different ways to do this. Finally, we talk about EEG, MEG modeling briefly, and really at the end, I want to give you an overview about software and the different tools that are used in separate pipelines. So structural preprocessing, we already saw T1 and T2 weighted images. We start with artifact correction. So what I'm taking some slides here or some images from people from the Human Connectome project. This is here Matthew Glasser paper from 2013 discussing the minimal preprocessing pipeline. They also have very detailed and very good, I would say, course material. So each year I think they have a course about the data acquisition and preprocessing pipelines. I have a link I think in the end of this talk. I can really recommend just to go through these slides. They have a couple of days of slides, a couple of hundreds, I think even more than 1,000 slides. One could go through, and they are pretty good. So one thing that they are doing is they begin to correct these images for gradient nonlinearities, and this is specifically important for their scanner, where the head is, so they have a modified scanner for their project where the head is slightly positioned outside the isocenter of the magnetic field. And so this is what happens here. So ideally there's this magnetic gradient, or this magnetic field decays linearly with spatial direction when you're moving away from the isocenter with Z. And this is what we would like to have, and this dotted curve is what we actually have. And these differences in the magnetic field cause then distortions. And as you can see here, if you're very close to the center, these differences is very low. So these artifacts are not so pronounced in most scanners. So it's not such a big issue if you, for example, in our data when we scan here at BCAN, but they have these artifacts here. So they use these gradient coefficients of their scanner to then correct these distortions. So what you can see here is this is pre-distortion correction, this is post-distortion corrections, and these red lines come from tissue segmentation based on this image. So they first corrected this one to this one, then the tissue segmentation on here, and then just plotted the region boundaries between different tissue types on both images. And you can see that there's quite some difference between this one and this one. And of course, since this is the magnetic field affects all images, or is used, the same scanner is used for all imaging modalities, this artifact has to be ideally corrected in all images if it is done. Otherwise, accurate alignment between modalities will be less good. Another big artifact is this bias field. So what you have in raw data, again, this is true for all modalities. I'm just showing it here for structural scans. What you have is that you have a very slow and smooth intensity gradient. Yeah. So some parts of the image might appear brighter than other parts. Here it's very pronounced. Yeah? And well, this is caused by imperfections in these radio frequency antennas. So this is also a technical issue or technical aspect of MRI imaging, which has to be corrected, obviously. Why? I mean, we as humans, we can easily still detect, okay, we can see, okay, this down here, this is white matter, right? And this is gray matter, and up here we can also distinguish between the two. However, for a computer, for different algorithms, this might get hard because it would expect the same tissue having the same intensity all over the image. Right, so white matter should here have the same intensity or the same gray tone up here. And I'll show on the next slide what the impact of this is. But usually we estimate then this inhomogeneity field. There's different techniques to come up to get at this inhomogeneity field, and then we can just subtract that here to get a corrected image. So different ways to do this. Within the human connectome project, the minimal preprocessing, they use the square root of, was it the ratio or the product of T1 and T2 images, because in T2 and T1 and T2 images, this contrast this inhomogeneity is similar. And yeah, with the simple calculation, this field can be calculated and then corrected for. As I said, the algorithms for segmenting tissues, well, there are several as well, but what they would assume is that the tissue, one tissue type has the same intensity across the whole image. And this is basically not true if you have this inhomogeneity in here, right? So white matter up here is pretty dark, maybe as dark as gray matter down here. So it would maybe, the algorithm would have trouble to identify this as correctly as white matter if gray matter is as white as over there or as bright. So we have to restore this, and we can take a histogram of the image, the voxel intensities. Okay, so on the y-axis you have the count, or how many voxels are there, and this is the intensity. And what you see here, this is pretty, like a almost like Gaussian distribution. No, not really, but very unimodal distribution. And if you correct for this bias field, you get several peaks, like one here, one there, and there. So it's a multimodal distribution. And what these algorithms do is they fit some kind of Gaussian mixture model or something. So this is the histogram you get from the image, right? So this is again image intensities. This is the count. And then basically there's 3 tissue types in there, or at least 3 with having different intensities. This is the cerebral spinal fluid, very dark, black. We have white matter, which is very bright, and then we have gray matter, which is in between. And now we fit 3 Gaussian curves with different mean and different standard deviation to this histogram, to this distribution. And we can use now this information to identify what each voxel is. And so basically we take out one voxel, this one here, which has an intensity of 17,000. Okay, so this has to be here. And then we can look, okay, how much, what's the probability that this is cerebrospinal fluid? And this is this blue circle here. What's the probability for it to be gray matter? Would be this one, still pretty low. And what's the probability for it to be white matter? Well, this is pretty high, right? And this is used together with other information and other algorithms, but is used to segment the image into cerebral spinal fluid, the white matter, and the gray matter. I want to say here already that this tissue segmentation is very important for the tractography. I'll show, I have another image later, but we use this to constrain our streamlines. What we basically want is we want to reconstruct streamlines only in the white matter. So the streamline algorithm is just stepping through the brain, right? And if by chance it ends up here in the ventricles, then this is an unplausible result and should get deleted. But more on this later. So white matter, CSF, and gray matter. This, well, here not including, should include also gray, the subcortical gray matter here. This is used to construct, to build the regions of our brain network model. The next step would be then to reconstruct surfaces. So if you have the white matter, you can dissect this into left and right hemisphere, and you construct the surface pretty simple in the first step just by putting 2 triangles on each boundary voxel. So a voxel is like a cube, and you put two triangles on the outer face of the cube, not going, not touching any other white matter cube. But this is a very in the first step it's okay, but it has very sharp edges, right? It's very blocky in its structure, so it has to be smoothed. And there are different algorithms, again, taking the intensity of voxel intensities into account and to smooth it to make it more look like what we have in what we would see in a real brain. Yeah, just here. This is basically I can already say it now. That's software using FreeSurfer. So FreeSurfer. I don't know if you are familiar with these different software tools, but this is done used for surface reconstruction and then also the subcortical segmentation here. Yeah, so this and this is very important because in our pipelines, or the way we are processing most of our imaging data right now, is we again here GLASA, right, the Human Connectome project, email preprocessing pipeline. We try to take as much advantage of their high quality preprocessing scripts as possible. And what they are doing is they are representing MRI data in this new SIFTI file format. So what they're saying is basically the gray matter, the cortical gray matter, is nothing but a sheet. We don't have to represent the cortex as a volume structure, but we can think of it as a sheet, which is folded according to the gyri and sulci. And why don't we just map the Why don't we just map the data which lies in the here between PL and white matter surface? Why don't we just map this the volumetric data to the surface, and then you get surface-based data within a SIFTI file format. Then you're not talking about vertices or voxels anymore, but they're talking about gray ordinates. And gray ordinates are basically vertices and voxels together. You have 30K vertices for the left cortical surface, 30,000 vertices for the right cortical surface, and then you have voxels representing the subcortical structures. And then these are the ones we talked about: amygdala, nucleus accumbens, brainstem, nucleus cauda, cerebellum, and both for left and right. Okay. So what are the benefits of doing surface-based data representation? Well, aligning them with an atlas or between subjects is a lot easier. Yeah. Moving around vertices on a sphere, on a sheet, is easier than in 3D volume, and you get less blurring. So most of the fMRI preprocessing steps and for statistical analysis, they also do blurring, and what this could lead to is that you blur across region boundaries. So this volume blurring doesn't take into account the true distance between two areas on the two-dimensional cortical sheet. So the distance from B to A in a biological or physiological sense is much larger because the signal has to travel all around the sheet here than its distance is in 3D. Right, and this could already be two different areas, and what you would do if you just blur in volume, you would mix the signal across region boundaries. And this is more prevented if you are using surface-based data for cortical gray matter, at least. There's other advantages as well, but these are, in my opinion, the two most important. Also, okay, this is less, it reduces the data amount, of course. You have less data to store. Yeah. The next step would be diffusion preprocessing. Again, we start with artifact correction. The artifacts and the structural scans were pretty light, so we talked about this gradient nonlinearity, which is present here as well. We talked bias field, which is present here as well, but other than that, the artifacts are not have not such a huge impact. Here, however, sorry, just need the time. Yeah. Here, however, we have to deal with a bunch of other artifacts as well. One is called Gibbs ringing artifacts. This occurs at sharp intensity transitions, between the different tissue types. This is basically an effect of the lower resolution in K-space in image. So when we acquire an MRI image, right, we have we have amplitudes for the different spatial frequencies contained in this image. They are there's for different spatial directions. You have the different amplitudes in K-space, and then you use a Fourier transform to construct such an image. Anyway, so basically what it's saying, we are not having enough samples here, not the frequency is not enough to really reconstruct these sharp edges perfectly, like the red, the black signal. Yeah, this would be the true image, and the blue one is the reconstructed one with our imaging technique. And what happens is that you have these rings here, or oscillations, damped oscillations, before and after sharp region boundaries. And then they can be seen also in the original MRI image. As you can see here, right, this is a sharp boundary here. Here it's pretty homogeneous, no ringing here, but along the gyri and sulci, where there's CSF, which is very bright, and then dark brain matter. And different algorithms again can correct for this artifact. As you can see here, then these rings are gone. This is one issue. Another one is eddy currents. So because of the imaging sequence we're using for fMRI or for Bolt, we induce currents because of the rapidly changing magnetic field. And these currents as well, they induce distortions in the image. So this is what should be. This should be the region boundary. This is what you then see for different setups of the diffusion MRI. FSL eddy correct. It's a tool in the FSL toolbox which corrects for these distortions. One that is also very pronounced and which is very dramatic in a sense is the epi-distortion correction. So different substances impact or interact with the magnetic field and then can either disperse it or either stretch it or contract it, depending on what the magnetic property of this material is. And this is something that also happens inside the brain, especially at region boundaries again. So it's very pronounced here, frontal lobe, and also in the temporal lobes, which is not seen here. And it leads to these distortions in direction of the phase encoding gradient. So here we are having a posterior to anterior encoding of the phase, and we should see a compression here, and then something like a stretching of the brain in the front. And if we reverse the phase encoding from anterior to posterior, then we have the exact opposite of the artifact. We have here this compression and a stretching here. Now, what we do to correct for this is either we acquire a field map which measures these distortions. This is, I think, mainly done, or this is how it's done in functional MRI. Here we're in diffusion imaging, we just acquire two images, two series of the images with reversed phase encoding. So what we can calculate from these two images is then like the mean something. Yeah? We see, okay, distortion in one direction and then distortions in the other direction, and we can use information from both images to correct for it and then get a well-aligned image afterwards. Now let's get to the fact about or to the technique of diffusion-weighted imaging. So what we do is we, as I said in the beginning, we measure the diffusion of water molecules in a certain direction. Yeah? So we can shape our magnetic field to just acquire information of water molecules diffusing in this direction. What we see is such a gray image, yeah, with dark areas indicating high diffusion, and bright areas indicating low diffusion in this particular direction. And we do this once for this direction, and then we do it again for another direction, and we might get a different image. Okay, the intensities here have changed. Now we do this for many different directions. And for each direction, we get such a gray intensity image. And then we use, where is it? Sorry. We use this thing here. So we take voxels in the gray image where we see very good diffusion in one direction, where it's nearly unrestricted. And we use this signal, which we get from there, to deconvolve the signal that we have measured in other voxels. So what does it mean? If in one voxel all fibers are there, aligned in one direction, then the diffusion of water molecules in this direction should be very good, right? There shouldn't be a restriction of diffusion there. This is the signal that we would acquire then. And in other voxels, you might have crossing fibers, yeah. And basically the signal that you measure in these other voxels is just a convolution of the fibers in that specific voxel with the response function, or the signal you would measure if all voxels were aligned perfectly. And this is what you get there. And basically this is the signal that you measure then in the MRI. And basically what we then do is, if we have acquired such an image, the gray intensities, yeah, this is what we measured. We go backwards. We know this, and now we compute this. And this is the fiber orientation distribution function. In the end, this gives us such a glyph, which contains the probabilities of fibers pointing in the different directions. This is especially important in areas where there's crossing fibers, yeah, to identify intersections between the streamlines. And we do this in each voxel of the image, and we get such a map. Yeah? So also because of noise and other artifacts, you also get such intensities here, very small. You can see them, these glyphs, even in cerebrospinal fluid and outside in gray matter. So we have to correct for this afterwards. But let's think about this for a moment. What does this information tell us? Well, it's the probability in what directions fibers are going in each voxel in the brain. But this is what we really want, is the streamlines, right? So how do we get the streamlines? Well, we take this as a directionality information, and then we step through the brain. Yeah, so this is such a glyph. This is a little older technique, this from diffusion tensor imaging. And basically such a glyph would indicate the streamline. Let's wait. Such a glyph, yeah, indicates streamlines in two directions, right? This one indicates a streamline, okay, only traveling in one direction. And now we have such a shape in each voxel of the brain. Here 4 voxels, and they indicate directions. And what we just do is we walk through this voxel and just follow the directions. And we do this systematically for all the brain in 3D. And what we get then are these streamlines, right? Not only in a plane, but in 3D. We do this systematically 1,000,000 times, 100 million times maybe. And finally, we get this tractogram, okay? And together with the cortical parcellation and the subcortical regions we had before, we group now these fibers together, and we get an abstract representation of the connectivity as the graph that we use in TVB. Now, as I said before, there's quite some biases in here, so we cannot only take the streamline counts as an indication for connectivity, because there are a lot of false positives, maybe false negatives as well, and the streamline count itself is biased as well. We get to this on the next slide. But one thing we do here to constrain our tractography is using the information of an anatomical image. So we have segmented the structural image before, the T1 scan, into gray matter, white matter, subcortical gray matter, and ventricles or CSF. And we have aligned these images, right? We corrected the distortions. So ideally there's no artifacts left in a diffusion weighted image. And we align them with the structural image. And now we use this information to say, okay, only a few of these streamlines are really plausible, and we will keep them and reject the others. So for example, if a streamline ends or begins or ends here in the, we start walking through the brain, right, anywhere in the white matter. So we can randomly start at different points and then walk in both directions. And if we hit the gray matter here, as described in 1, then that is plausible, right? Because this is what we really want to reconstruct, the fibers, axons from one brain area to the other. So we keep this fiber 1. However, if the streamline ends in a ventricle, this is implausible. Yeah? Because there's some randomness, we're using probabilistic tracking here. Sometimes it happens that we step into a ventricle, and this is implausible, so reject this one here. If your streamline enters the subcortical gray matter, that is fine. Keep it, but don't let it exit subcortical gray matter again. So this would be unplausible. So what this allows you is to also have more. It shouldn't that the streamlines don't only end on the surface of your subcortical gray matter, but also go deeper. So you have some more information of the inner subcortical gray matter connectivity. If streamlines reach the end of the field of view, that is fine too. Yeah, because this is just a spinal cord here, brainstem, so this is plausible. If, however, streamlines end somewhere in the white matter, that's implausible as well, because we assume them to go from one gray matter area to the other one. Why should streamlines actually end in the white matter? Well, there's certain already constraints when stepping through the directions in the white matter, that very sharp angles are prohibited. Yeah, so there's nothing that the streamline should always very smoothly change directions. Yeah, there's nothing like a 90-degree change in one step. So this is implausible as well. And if streamline tractography ends here because of such a criterion, this is also implausible and gets rejected. So what is the result in the end? Here we're looking at one brain region a little more in detail. So we're actually using two different techniques here to construct the directionality information for this specific excerpt of the brain. Here, this is done with this deconvolution approach I just said. And you can identify different, you have these glyphs, right, also representing crossing fibers, or potentially crossing fibers. Here you have these diffusion tensors, which basically just show in one direction, and they have difficulties to identify these crossing fibers. So what do we see here? Well, this is the approach without anatomical constraint tractography. Yeah? And these red dots indicate a termination of the streamline. So blue, green, and blue and green indicate and red here indicate the direction of how these streamlines are flowing, and the endpoints are indicated here. So if we look at this image again here, this section actually, so this is the ventricle right there, and this section here is actually right in white matter. So why is it that some streamlines end there? Well, this is the reason I said before is they terminate there because there's implausible curvature of the streamline. So they end there, several others end here at gray matters or even within gray matter. And now we use anatomical constraint tractography on this one here, and these streamlines get deleted, right? Because they're implausible, they're ending inside white matter. And what we remain with is this this image here. You know, there's empty spaces here because these streamlines were deleted, and there's now an underrepresentation of these fibers in here. So it's quite an issue, quite troublesome that using this imaging technique or these diffusion tangles, tracking and then deletion, leaves us with less plausible than images. So these are not plausible, but then also these holes in here are also not plausible because there would be actually fibers. And why do they end here? I said they're doing an unnatural curvature of the streamline in this step. This is caused of the fact that here they're crossing streamlines. And this technique isn't able to identify this crossing of the streamlines. However, if we use this deconvolution approach I explained before, we get such an image, again with many different endpoints, even within gray matter up here, even endpoints in the ventricle, which is also implausible. So we use anatomical constraint tractography, and what we get is a nice image, only endpoints on the gray matter boundaries, which is perfect, or which makes sense. And we also don't get this underrepresentation afterwards because we were able to correctly or potentially correctly identify the crossing streamlines. So if you are the message I want to convey here, anatomical constraint tractography has an impact, and it should be used with techniques that can deconvolve the signal into different and to have an impact of crossing fibers. Another filtering or another correction algorithm is spherical-deconvolution informed filtering of tractograms. So imagine this is the image you've acquired and then deconvolved it, and what we can nicely see here, we are only in the white matter, and we have these glyphs only in the white matter. That is good. And now we do tractography on this one, and we reconstruct streamlines. And we do this millions, so it's really densely packed here. These are the green lines. These are all fibers inside white matter. And now we take this image as if it were the real brain, and we are measuring now these glyphs on this image, right? So here what's missing would be actually the ground truth. The biological streamlines would be here, and from this biological ground truth we are measuring this signal, and now we're using tractography, as explained before, we are reconstructing the fibers, and we get this image. And now from this image we measure this diffusion signal. Now we can see that some areas are very, very overrepresented. So some areas get very big glyphs which weren't there in the first image. And this is because biologically each axon has also a cross section, that it occupies space in 3D, and there can only so many streamlines fit into one voxel or into one area of the brain. However, with these streamline techniques, you can fit as many streamlines as you want in a certain brain area, and this then gets unplausible, right? So we have to correct for this, and there are two approaches, Zift and Zift2. Basically what they're doing is they're assigning each streamline in the brain, I'm now talking about Zift2. Each streamline they're assigning a cross section. So how wide is the streamline? How much connection does it, or how much weight? This is important for our connectome construction. How much weight does this streamline contain? And then if we reconstruct from these streamlines the diffusion signal, it's much more closer to what we started with in the beginning. Yeah? So care must be taken when trying to interpret these streamlines as real connection strengths. So yeah. Functional preprocessing. fMRI measures the blood oxygenation in brain areas, right? And we think that there's a relation between brain activity in some area and then an increase of oxygenated blood in that area. So if we measure blood oxygenation in the whole brain for a long time period, then we might get a signal that tells us something about the activity of the brain. And this can be done on rest. So people just lie in the scanner, have their eyes closed, try not to fall asleep, don't think about something particular, and then we measure activity, or we give them certain tasks. They have to, I don't know, calculate, do something, a motor task, remember something, mathematical task, and so on and so forth. So there are different modalities there. However, if we acquire the brain, the whole brain volume, every, let's say, this is TR, right? Repetition time, let's say every 2 seconds, or these very advanced scanners or new sequencing new image protocols from HCP, they acquire one every 0.7 or 0.8 seconds. So you get one volume for each time step. And one issue there is that patients move over time. So you have to realign these brains all together to make then in the end comparable, or for asking statistical questions, or for having such that each voxel represents the same brain area or the same position in the brain across the whole time, right? And this is what's just depicted here. You begin here maybe, and then you move your head to the right and then to the left, and all this has to be corrected, has to be realigned and again. However, there's even more issues with this than just realigning them, because voxel intensities, depending if you move through the magnetic field, then change their past or the futures, then depending on their on the history and depending on whether you were excited before or not, has an impact on the signal. So we also use then these motion parameters for regression analysis to clean even further motion-induced noise in the end. So these are motion parameters, yeah. This is then the TRs, time points. Yeah. I think 1,400 was 20 minutes, could be. Yeah. And this axis here is either degrees, rotation degrees, or millimeters, I think. Yeah, millimeters. Now, why do we have 6 timelines here? You have 6 degrees of freedoms, maybe, mainly. You can either translate your head from left to right, up, down, front and back, or you can rotate along the 3-dimensional axis. So you have 3 motion parameters, and what you can see here, which is pretty interesting, is that you have these bumps, right? There's 1, 2, 3, 4, 5, 6, 7, 8. Now, why is that? Basically, this was a study with 10 task blocks, and what you see is here people concentrate during the task. They get excited or very focused. They move a little bit, and then during the rest block, they fall back and relax. So that's why you have these like these spikes in here for the task. Yeah, important to consider if you later search for brain activity. Sometimes what you see is just not related only to the task, but also to the motion that is correlated to the task, as you can see here. Another thing, slice timing correction. Because we acquire the volume, let's say every 2 seconds, the whole brain volume, but separate slices, as you can see here, 1, 2, 3, 4, 5, 10 slices, let's say, they are acquired at different time points. Yeah, we acquire one slice after another, and then there is some delay between one slice and the next. And even so, the underlying signal in the brain region was the same. Yeah, you would get something like this. Let's imagine the brain activity in these 2 slices is the same, but due to the delay in the acquisition, the measured signal in the end is slightly delayed here in slice 10 compared to slice 9. But we know when these images were acquired, and we can interpolate, or when these slices were acquired, and we can interpolate the steps in between and then just shift this one back. This has been very important in former days when TRs were even longer, like 3 seconds or 4 seconds. Now with 0.7 seconds, this is not even considered anymore in the human connectome project pipeline. AP distortions, similar as to the ones we had in the fusion-weighted images. We use a field map. There are different ways to measure field map. Span echo-based or gradient echo-based field maps. So basically what they indicate here is the distortion of the magnetic field in different areas. What you can see here, right, these are the sagittal, axial, and coronal slices of the brain, and you see here that in temporal lobes and frontal lobes, these dispersions are strongest. And this is what you can again see here as well. Posterior to anterior phase encoding. This is now MNI, fMRI signal, posterior to anterior. And you see again the distortions here in the frontal lobe and also a little bit in the back. And you use this information to unwarp this. Basically you get a good shape of the brain afterwards. Again, this is very crucial if you align this with your structural scans, because you have the information of the regions in your structural image, but you have the information of the function in the functional image, in the fMRI. So if you have a really good alignment between them, then you can really infer what is the function of a specific region, or how is it behaving over time. Now, this is those were some artifacts in fMRI. However, there's still temporal noise in it. What do I mean with temporal noise? Well, there's certain components also interacting the times, also distorting this time series, which are not of neural, neural origin. So there are several other components in the signal we actually measure, which are not neural and which we are not interested in, or which we don't want to analyze. And actually they make up quite a lot of the signal, and we have to remove them before. So there's motion, as I said before, which induces, especially at the edges, induces induces noise into this time series. This is crucial for, especially when we're interested in cortical activity, right? There's white matter activities which we're not interested. Motion again, now more pronounced frontally. Cardiac pulsation causes, cardiac and respiratory pulsation causes artifacts. Sometimes this is also represented in the ventricles, so in the cerebral spinal fluid. And these are here, for example, the sagittal sinus back here, which adds to the measured fMRI signal, but is not neural origin and is not of interest for us. So what we do is we use independent component analysis to decompose the signal into independent component, as it says. So you can think of this as something like 2 or 3 people speaking, and we have 2 or 3 microphones in front of them recording. And each microphone will record all of the 3 speakers, but with different weights, right? So the one that is positioned closest to me will record my voice the loudest, but one that is positioned here will also record my voice, but maybe the speaker next to me, his voice will be recorded stronger. Yeah? Imagine that all 3 speakers are speaking at the same time. And ICA then helps to, takes the signal of the microphones and then entangles the 3 speakers. And in a perfect, under perfect conditions, you would then get the voice or the audio signal of each speaker separately. And here, the speakers are motion, white matter, cardiac, sagittal sinus, respiratory, and neural components. Yeah? And we want to disentangle what we recorded with our microphones into these different components and then remove all of that we are not interested in and only remain with the neural signals. And that's not only a spatial pattern that is interesting or that's important, and we can also decide into good or bad components based on their temporal features. So good components have oscillations in a very, so this is the time series of the component on the left, and this is a frequency spectrum, and you see they have very slow oscillations. Yeah. However, bad components have frequency content also when in many very faster components, as you can see here. And this is called ICA Fix. Yeah, this is a tool of the FSL toolbox again, which does the ICA decomposition and then labels automatically the different components into good or bad and removes all bad components. You might ask, how does it work automatically? Well, it has to be trained first. There are classifiers coming for default data, for the high quality data of the Human Connectome project. From my experience, it makes sense to go through, let's say, some subjects of your study and hand label components by yourself. So you have to really look at the spatial distribution of these components. Yeah, we have this here. This is, for example, one component. You see these spikes again here. This is from the same study where we got the motion parameters before. And we go through all these components. It could be like 100 for one subject. And we hand label them good or bad, good or bad. And then Fix takes these hand labels or this information about the components and each component and trains itself. Yeah? So it takes features like the spectrum here, the where is activity base? Is it on the boundaries? Is it somewhere in the middle? Is it very diffuse, or are these big blobs? It takes these different informations, trains itself, and then it can be applied to the next 30, 50 subjects of your study without you being required to hand label them. This is an artifact component. Why? Well, it is pretty slow here again, but it has very sharp edges, right, very sharp spikes here. This is again these task blocks. Yeah, there's no, also the spatial component doesn't really look good. If it was neural, we would expect like bigger blobs somewhere, corresponding also to known resting state networks or task networks. This, however, is better. Yeah, it's smoother here. We don't have this. We don't have this sharp transition, for example, here. This looks much better. It's the same subject and just another component. This looks pretty much like the default mode network, right? We have activities here, parietal, cortex, frontal cortex a little bit. The oscillations, lower frequency, and so on and so forth. So we can use this. Of course, it requires some manual input in the beginning, but it's definitely worth the effort. Another technique for this as a toolbox is called ICA Aroma, which does everything automatically. You don't have to specify this by hand. It tries to estimate motion components based on 4 features, based on how much is the activity around edges. If we go back, we can see here motion is outside of the brain, activity outside of the brain. So this is one feature. How much of the activity of the component is within the cerebral spinal fluid. how does it correlate with motion parameters and high frequency content. And then you can use different machine learning algorithms again to classify the components into good or bad, and finally also again subtract these bad components from the signal. Something else I just want to mention here as well is global signal regression. The approach in the end is similar, that you have some time series that you consider is unplausible or that is a noise, and you regress this out of the signal. So in ICA this was the artifact components, which we regress out of the signal. Here global signal regression is meaning that we take the global signal of the brain. So we just take old brain voxels, average them, and use this as a signal to be regressed out of the other brain regions. The assumption behind this is that noise is present everywhere. However, neural activity is very regional, and if you average across the whole brain, you get this. You get out the noise, which is present everywhere, and you can subtract this from the rest of the signal, and you won't subtract regional activity. However, this is not so clear in practice, and very big neural activities or big neural networks might also contribute to the global signal. So actually in the global signal you have neural components and artifact components, and this might lead to the fact that you also regress out actually variance or information from your data that you actually want to analyze. And so this is controversial. We're not doing this. Just wanted to mention that people are doing it, or it has been done maybe. Another effect that this has is it moves your, especially this is important for functional connectivity analysis, if you then correlate the voxels with each other and you get your functional connectome, it shifts the correlation coefficient, as you can see here, from positive values more to the center to negative values. So yeah, it's argued that this technique, global signal regression, might introduce spurious negative correlations in your functional connectivity analysis afterwards. Okay, now a few words about cortical parcellation. There are different brain atlases out there. How are we in time? 10:30. What does it say? 11:20, right? No. Yeah, 11:20. Okay, so we have 40 more minutes. Cortical parcellation, there are several brain atlases out there to group the cortical surface into different functional areas. Some are volumetric based, maybe defined in MNI space or on some template. What then people do is they morph, they register the subject structural scan with the template, and then just copy the region labels from this template. As I said, it's pretty hard to register, or it's harder to register in 3 dimensions than in 2 dimensions. This is why people now consider more or we are, let's say, we are considering more the cortical parcellations that are surface-based and which are using multiple modalities. So not only anatomical landmarks, but also other imaging modalities to refine or to get a more accurate subject-specific parcellation. Where does this all come from, or where does it start? Well, it started with Brodmann more than 100 years ago now, who sat down and microscopically looked at all the different brain regions, or had sections of the brain from multiple different areas, and he created a brain atlas with, I think, 36 regions. These numbers here are higher because some are only defined for non-human species. And he had a look at the cortical columns, or the structure of the gray matter, cortical gray matter. And he saw, okay, that there's different cytoarchitecture between areas, and he used this as a feature to classify different areas. And well, he created an atlas. This is what many people since then have used and refined and added other areas. So this is where it all started. And well, the main property here was the cyto architecture that has been used. So how do we map this then to the subject? Well, let's go back again. We can use certain anatomical landmarks. So some gyri and sulci are very pronounced, and you can find them in almost all human brains, right? And you can say, okay, based on these landmarks, you can say where a certain area should lie, and from there on map this to the subject brain. However, we know that this cortical folding is pretty robust for the main gyri and sulci, but if you move away from these primary motor, visual, auditory areas, gyri and sulci become very subject dependent. This, for example, for twins here, and even in twins you can find cortical folding that is different. So the idea is that only based on anatomical landmarks, the registration of the atlas might be a little bit inaccurate. And also what has been shown here, that this is post-mortem analysis from 4 subjects, that the brain areas not really stop at anatomical landmarks, so they go across gyri and sulci, which again then adds constraints to this process of using just anatomical landmarks to map to the subject space. What other people also said then, okay, not only on, this is work also 100 years ago already from Cecile and Oscar Vogt, who studied myelin content or myelo architecture of the brain to separate between different regions. Here's a microscopic picture, and these are the regions visual 1 and visual 2. And you can see the myelin contact here stained, and there's a sharp boundary here where some architecture, like this, several layers architecture ends and goes over into the next one. So this is something that also could be used to distinguish between brain areas. Now this is all this, and this data comes from microscopic sections. However, how do we do this? How can we make use of this myelin content and in our subjects where we are dealing with living human beings and we only have MRI data? Well, it turns out we can actually measure myelin content in the cortical sheet. So myelin shows up in the contrasts of T1 and T2 images, and just the ratio of the two already predicts quite accurate where myelin is based. And so this is the T1 image. You can see the blue line is the PL and the green line is the white matter surface. And here this is T1, this is T2, and now you lay them on top of each other and take the ratio. And you see then here the myelin contact, and red means high myelin, and gray or green is lower myelin. And so you can, what I want to show here is that you can actually measure myelin in the cortical sheet with MRI as well, and use then this information to delineate regions or construct region boundaries in your subject, which are more accurate than just taking anatomical landmarks into account. This is then such a myelin image, a myelin map on the cortical surface of a human. The top row is humans, left and right from medial and lateral view. And what you see is we have much myelin in these primary sensory and motor areas, in auditory and visual cortex, and less myelin in higher cognitive areas. And what's shown here is for a chimp and for a macaque, and what Glasser showed here is that actually this myelin can be used to identify homologous regions across species. So also they have their sensory motor areas highly myelinated, their visual cortex, and so on. So we can use myelo architecture for estimating region boundaries. This is one thing. But actually what the Human Connectome project does to even further increase accuracy is they take many many modalities into account to delineate the regions. So this part A here is the myelin map we just saw, right, with some regions identified on it, region 55B, and the region boundary in dark around it. This is task fMRI, so this comes from, this is the activation pattern, right? These areas are activated for this specific task, and the task was story language task. So they were basically taught a very short stories, a few sentences, and then had to ask a question. And what you can see, okay, several areas, different areas activate, and others don't. So this information is also included to separate between region boundaries, or between regions. Functional connectivity is one measure. For example, if you take this area here, PSL, and do the functional connectivity of this, so you look for other spots in the brain that correlate with the activity in this area. You see, okay, in the surrounding it lights up yellow, here, there, in this part. Yeah. So this seems to be some functional network. On the other hand, if you are using activity back here and correlate this with other regions, you see activity in the visual cortex, and up here the neighboring neighboring area of this area 55B. Okay, so also functional connectivity maps can be used as a modality to identify different brain areas, together with myelin, task, functional connectivity, and also topographic information about the visual cortex, which I didn't cover here yet. So we have this information, and what is then done is we compute the spatial gradient. So we are not from the areas where we see there's very high myelin, for example. We use as a criterion to construct a boundary to the bordering regions is the spatial gradient. So this is what's then left here. For example, task fMRI. You see this red circle, or this red boundary here means that there's a change in this statistical map, and they are used for dissecting the brain into different areas. Maybe here a quick example for the motor strip in more detail. We still use cortical folding as a first measure. We're not completely rejecting, or Glasser and his colleagues are not completely rejecting that cortical or anatomical landmarks wouldn't indicate the different areas. And this is the first step. Then we use myelin. And what you see here is the different areas, 1, 2, 3, 4, and the boundaries in black. Yeah. And you see myelin. Okay, there's already a difference in myelin between these first three and then the last one here. The cortical thickness plays a role, right? It's also different between the different areas. These ones are pretty thin, actually. This area 3 compared to the rest. Functional connectivity gradient, well, in this case, doesn't seem to play such a huge role. And this is, I think, a task fMRI again. And based on this, you can then construct these probability maps where, giving you an estimate of how likely it is that this brain part is some area of the motor cortex.
Well, can I ask
Yes?
you something? Can we choose some
It's the next 2 slides. Maybe we'll do this. I'll answer this question in the next slide. Okay? Yeah. So how is now this information technically done to separate, or technically used to separate between brain regions? Well, in this Glasser paper of 2016, they used a semi-automatic approach. So first they had neuroanatomists sitting down and drawing these regions. So they had these gradient maps, and from their knowledge, from the literature, and from the different modalities, the gradients they took and draw the regions by hand. They had a very brief, then they used an algorithm then to smooth these hand drawings, right, a little bit to optimize this into the other gradients. And then this was taken as a template. Many subjects were averaged on this or registered to this, and finally they also constructed a classifier, which to my knowledge hasn't been released yet. But this classifier was then used taking the different modalities and would then say, okay, where's the different areas in this particular subject? So this is one technique. You give all these modalities into this classifier, which tells you the location of the different brain areas. Another way is to have this template on the surface. Yeah. So we're using a tool here called MSM, Multimodal Surface Matching, by I think her name is Emma Robinson. Also a tool in FSL, which does surface registration. So originally you would register the volumes, correct? So you have a grid of voxel intensities again, and you would move them around such that the intensities align again better. So this is what was done originally or traditionally. On a surface, you move around vertices, which is easier because it's in 2 dimensions, but the principle is the same, right? This is one voxel point here, or one grid point, and you move it to the direction of x, where some cost function is best minimized. Here it is similar. You move then, but just on the vertex or on the vertices. So you get basically what this is all done on a sphere, but you have in the beginning you have of course the cortical surface of the subject, and you inflate it. It's like blowing up a balloon, and you get a surface in the end. And on the surface you have the different modalities, because we were saying we map the myelin onto the surface, right? This is what this was about. We map myelin to the surface. We map statistical maps of task FMI onto the surface. We map functional connectivity onto the surface. And now imagine all these surfaces are laid on top of each other and inflated to a sphere, like this one here for myelin. Yeah? You get a sphere here, and each vertex on the sphere has some myelin intensity, some functional connectivity intensity assigned to it. And on the other side, you have the sphere of the template with the same metrics, the same modality also specified. What you now begin to do is you move these vertices around such that the modalities of the subject and the template align best. The steps then here, and then you can move this back into the anatomy, like deflate this, but the vertex now have changed, and you can then just take the parcellation, which is defined on the template sphere, template surface, onto your subject. Now the question was that all these modalities have to be taken into account. No, but then you lose accuracy, of course. Yeah, you can also, what we did, for example, with our Alzheimer, with the data from the ADNI study, we also once just took like cortical folding, myelin maps, and cortical signals as a measure, and then did this approach, this surface matching, only with these metrics. So it can be, ideally you have all of them, but it can be done with less. With then you have to take into account the inaccuracies that you are getting.
Well, it might not be inaccuracies, it might be what you want to do, right? Because, for instance, you might be interested in some functional aspect or not.
Yeah, and okay, with what you want to do here for functional region identification for TVB. That's, yeah, you're right. But yeah, this tool is like pretty flexible. So it wants you to give 2 spheres and the metrics on the spheres, and then it does the matching. So we end up with this human connectome project's multimodal cortical parcellation. So this is subcortical part, right? This gray is not of interest, or is covered by the SIFT file format and the volume, the subcortical segmentation, the volumes. These are the cortical regions here, color just indicating how much they correspond to any task fMRI, or not, task positive, task negative here. And they have 180 regions for left and 180 regions for right, plus 19 subcortical regions. So we come up with around 379 brain areas. No. Oops, sorry. Okay. Just briefly, EEG forward modeling. What we assume is that, how do we think that EEG is created? So there's a current flowing in the pyramidal cells, which is very, very small. But because the pyramidal cells align and they're perpendicular to the cortical surface, these currents add up, or the electrical field adds up, and it can be, it is projected to the cortical, to the scalp, and can be measured with electrodes. Similar for, this also creates a magnetic field, of course, and this can be measured with MEG. We in our group, I think, didn't do MEG ever, but mainly EEG. Now it is, I wouldn't say pretty straightforward, but it is possible to compute a forward solution. So we have the cortical surface, right? We also know where's the skull, what's the surface of the skull and what's the surface of the skin. We assume different conductivities for the different tissue types. And we know where the dipoles are positioned on the cortical surface, and also the normal vectors, right, serving as the orientation of the dipole. And now we can calculate how much would each dipole project onto a specific location on the scalp surface. So we know where are the different EEG electrodes, right? We are mapping them from the real human being with different techniques, or we just use the default data, default EEG cap, that's possible as well. So we stick them on there, and then we compute how much would each dipole affect each electrode on here. And well, this is then basically this lead field matrix, which I announced in the beginning, like a table or matrix, right, where you have rows which correspond to the EEG sensors, and you have columns, one for each dipole on the brain. And if you, so each vertex on the surface would be one dipole, and you can obviously, now for region-based simulations, we just group them together, we average them, and then use the use this as an we use this for projecting the neural activity which happens on the region level onto the EEG sensors on the sensor level. The opposite is a little more tricky, because so this would be the forward model, and then the inverse solution or inverse problem would be, if we measure activity up here, how is this transferred to activity up there? And there you have to put some more constraints on the mathematical equations, because you're not having enough sensors here, and with not high enough, or because the activity is blurred across the different boundaries, tissue types. What you're measuring here isn't spatially resolution enough to estimate these many possible dipoles within the brain. So that's another topic. Maybe Julie will talk about this when she's doing epilepsy. Yeah, because there this comes into play. So just briefly in the end, I want to, oops, what happened? briefly in the end, I just want to give an overview of what different software tools and software packages we are using. I think FreeSurfer and FSL should be familiar with people who are working with neuroimaging data. FreeSurfer doing this brain surface reconstruction mainly. It's called recon-all pipeline in there. Takes a lot of time, 12 to 24 hours, depending on your computer resources. FSL, well, you can run this in parallel a little bit and it's get faster on a cluster. FSL, many different tools in there, many different options to process data for fMRI, but also, as we said, the surface matching, ICA figs, ICA decomposition. Connectome Workbench, a tool from the people inside the Human Connectome project, especially to work with the SIFT file format. This is very helpful. Of course, some tools here, especially machine learning, rely on these programming languages, and sub-packages within there are Python, MATLAB. So it's pretty, these tools up here, they're then mainly called or organized using shell scripts. So something like bash comes in handy here when you're working with these. Here, it's a little bit of knowledge for Python or MATLAB will be handy as well. Matrix, the software we are using for streamlined tractography, has all these advanced artifact correction methods, has these different streamline algorithms, this anatomical constraint tractography, as well as after tractography, the filtering of the tractogram. Brainstorm and MNE are different software packages to use to construct the forward model or inverse solutions for your MEG or EEG. I can really recommend to check out the website of the Human Connectome project. This is the last course that happened, and some of my slides and some material were taken from there as well. As I said, very helpful, very in detail. And also they're very helpful when having questions, technically questions about data, software, but as well content-wise when discussing different preprocessing steps. So they have a mailing list, which is very good, I would say. These papers describe from 2016 describe the multimodal parcellation. 2013 describes the minimal preprocessing pipelines. These scripts can be taken from GitHub. Then one has to take care of installing all the different dependencies, the software packages, which I mentioned before. Yeah, they especially this. Sometimes it's trickier, at least from my experience, if you want to install all of these on a cluster where you don't have root access. However, there's also now, or they're working on a container, which I haven't used, I have to say, but seems to run, which is Docker or Singularity and comes with all the software packages pre-installed. So this might be easier if you actually want pre-processed data. But as I said in the beginning, there are many, I'm mentioning this here because we are using a lot of this pipeline right now, but there are many pipelines out there. As I've said in the beginning, Michael Schörner, 2015, did one. Our colleagues from Marseille did one, especially specifically designed for TVB. What HCP doesn't do is the last step to convert everything into TVB format. These pipelines also don't do the tractography yet, so this has to be added in order to make something out of this in TVB, but do the minimal preprocessing very good. One thing I want to mention is bits. So we had in the beginning this image where we come from raw data, which is mainly DICOM, and we want to construct these brain network models. And bits is a specification to store data in a specific folder structure. So DICOM gets converted into Nifty using different tools. And bits then would say, okay, how do you store anatomy data, structural data? How do you store functional diffusion-weighted data in a way that is findable, reproducible, understandable for other people? And one benefit of organizing your data, and it is a little work in the beginning, you have to read in the specification, but one benefit of doing this is that you can then use bits apps. So there are people who created Docker containers, either Singularity, Docker, and they work on this folder structure specified by bits. And then it's pretty easy at some point to just acquire data, store it in bits, and then use some Docker which has been tested, which has been validated and has many functionalities, and to just move your bits dataset into this container, and you get an output. The aim here is to make this pre-processing and image analysis more reproducible and more robust, so that the neuroscience, neuroimaging community takes similar steps when preparing their data. Here I'm showing you just two screenshots of the HPP Collaboratory where you received an invitation to. We also constructed one pipeline recently based on these two bits apps, Matrix 3 Connectome, which does structural and functional preprocessing, gets you the structural connectome, and fMRI Prep, which does fMRI preprocessing, gives you the functional connectome. And then we wrote our own little Docker or our own little container in the end, just to convert the output of the two into something that TVB can work with. And we installed everything here or we created a Jupyter notebook for you, or for anyone in HPP, that leads you through the different steps to execute this container, and then our converter to TVB container. It has a lot of explanations in there, so this can be checked out as well. Now to summarize this long session, which we're almost done, is this video, also created by Misha in his paper from 2018. So what we do is we scan the brain, we scan the subject in the MRI. And this is what we get. We get a structural scan. We reconstruct the cortical surface, and in the white matter, we reconstruct the fiber tracks, right, with the different techniques discussed. Now, this is what we get, and on the cortical surface, we define region mapping or cortical parcellation into different areas, such that we can group the streamlines into edges that connect the different brain areas. And this is in the end our abstract representation of the brain as a three-dimensional graph. And with this I want to finish and thank you for your attention. If you have any questions, please feel free to ask. Otherwise, we're having the lunch break now, right? Coffee break, morning break.
I have done this for epilepsy like 30 times, right? And I still don't feel that I can look at an image and tell if something is wrong or not. So my question is, how easy is for the non-expert user to use one of these pipelines without really having to put his hands on the images, correct an error, or find out an error? And if there are such errors that usually, I guess, come because of bad quality of images, could you identify the steps where more often one needs to put some more expert input there?
So I would say if you identify image, or if you're dissatisfied with the preprocessing pipeline, at some point you will have to get your hands dirty and take into code and also the images. But a lot of these pipelines also provide or create quality control images or quality control assurance something. So they print you like JPEG or PNG files where you can see, okay, how well did alignment let's say, alignment between structural and functional. How well did it work? How well did these distortion corrections work? So some of the pipelines offer this capability. Your second part again was the...
But how expert do you need to be to see this PNG and identify there is a problem? Okay, the alignment I know it's very easy because you just see two lines.
Yeah.
But different artifacts, I don't know. I'm asking where is it?
I mean, the BITS apps, they're trying to make it as easy as possible, or they're trying to make this as easy as possible, and such that you wouldn't have to check that in the end, that you can, or at least the aim is to have this reproducible workflow. But I would definitely say you should be aware of all the steps that we're done. It's not a plug-and-play solution. It's never. Yeah, you should really know what these pipelines are doing, and if you really want to include these steps, because there's such a variety of different possibilities. Yeah, it takes some time to get this knowledge, to get more insight into the different steps, but I think it's worthwhile the effort. Yes?
So it's a related question, but I'm wondering for these pipelines, how much tuning of parameters is required by hand per subject or per data set?
Right. And you also ask about identifying the problems and where they come into play. So the biggest problems, I think, are in the beginning, misspecifying the metadata of your images, or at least that was when I started, this was my experience. That, as you say, they take a lot of parameters and input, and a lot of come from the raw data themselves. So you have to specify, for example, phase encoding directions. Which was it? Yeah, the different scanning parameters like dwell times, echo spacing, and so on and so forth, which has to be tuned not only in the processing pipeline, but also before that in already has to be discussed with the MR technician, I would say, to get the best out of your specific MRI scanner. The pipelines themselves later on can be tuned as well, depending on what type of pipeline you are using. If this is like an all-in-one product, or if it's separate steps, there's a lot of tuning in there. And I know from the HTTP people, they have their high quality data, and they try to test a lot of those different settings and try to come up with the best possible way. But yeah, I would say that the degrees of freedom you have are pretty huge and have a big impact on the preprocessing results, especially for fMRI. As I said, there are different denoising approaches, right? And anything based afterwards, then functional connectivity, FCD dynamics. Also, if we are fitting our simulated data with TVB to these empirical data, can be quite affected by the way you were preprocessing your data. So it's definitely an issue. Yes?
My question is, you have shown a picture of the multimodal surface and every region with a high dimension matrix with the multimodal, such as structure or the function connectivity. And then if you have already the structure from the GTI, and in every node you know the connection and the strength connection, and you would like to input the mappings, also the structure, and you also input the fold or fMRI.
Mm-hmm.
So how can you combine how can you evaluate the multimodal, and or another how can you input the data to the TVB, and how can you evaluate the multimodal?
Okay.
In general, if we
I get it now.
fMRI, we use the graph series, so the structure topologic is not the same with the structure. Or if you like to use such as the window shift for the dynamic every node, there will be some there. How can you combine the structure and function together?
So here, these multimodalities are used to identify functionally distinct regions, right? We use it to separate the cortical surface into different functional units. That's the main goal. Diffusion weighted imaging is a side. We then do the track tracing in diffusion images, yeah, and we then take the parcellation, put it on top, and we get our structural connectivity out of this. The multimodalities are not in this way mapped already into TVB simulations. Yeah? At least for now, no one did it. But I think people are now beginning to work on this as well, using, for example, myelin content. Yeah? Also in TVB to have a prior for certain parameters of your model. Yeah? And this then again generates a refined TVB model to simulate, I don't know, EEG fMRI, which can be connected to.
In general, how can you evaluate the fMRI in the 3D?
The simulated fMRI, you mean? Different metrics. Most people just use very simple, just correlate functional connectivity matrices with of the simulated fMRI with the functional MRI. You can use graph theory on your simulated functional connectivity and compare it with the empirical functional connectivity. That is what some people did. They also use, for example, in the mouse simulation, you used functional connectivity dynamics. So you looked at the temporal aspect of the functional connectome. How does the functional connectome change over time? This can be fitted to empirical data as well. This is your question? Was it? Okay. Good.